My name is Tatyana Goldberg, as some of you already know me. I did my PhD basically on this problem. This was the main topic of my thesis, and the method that I'm going to present today that started as my master thesis project actually, that then got expanded into a whole PhD thesis. But the work presented here was done within half a year. If you are interested in doing a master thesis with us, then you can get an understanding of what the scope of such a project is. And I can tell you for sure that this is like super cool what we're doing when we develop such projects, and it's like really fun, and you know that this is something that's like really useful for people as well. All right, so localization. Okay guys, what is subcellular localization? Can someone summarize it? With own words, like what do we mean when we talk about subcellular localization? Just try. Yeah?
Find out in what compartment the protein is localizing itself.
What is the compartment?
For example, the nucleus, or is it a cytosol?
Is extracellular space also a compartment? Good job. Okay, and why is it interesting for us? Why do we want to know where it's localized? What's the reason for asking this question? Someone else?
The localization has something to do with the function.
Why?
Sometimes.
How? Yeah?
I think there are some functions which, for example, are only executed in the nucleus. Like
Totally, yeah. And localization is actually an aspect of a function. So if you remember this Go tree, then localization, that's like 1/3 of the tree. This is one of the 3 hierarchies of gene ontology. Okay, so eukaryotic cell, we have different compartments. Each compartment performs its own cellular function. Of course, this is only a schematic representation of a cell. In real life, it's like super packed. The cell is like all the compartments there. Well, some are larger, some are smaller, but there is as good as no space, because if there is not a compartment, then there are proteins. And you can imagine how the cell inside looks like. If you remember of these videos of these crossings in Asia, where you have like a mass of people going this way, and then a mass of people going the opposite way, and then the mass of people going this way, and like it's really many people and stuff. And this is how in the cell it also looks like. It's like they all really shoulder to shoulder to each other. So it's never just as pretty as here. Okay, so proteins, as you all know, they have many different functions. As a matter of fact, what proteins do actually makes us alive. I'm sure that you all know that proteins, they can maintain the DNA, they pack it, they protect it from damage, from the outside damage. Our ribosomes, they help to DNA to be translated. Collagen, that's a protein we find in our bones, in our muscles, in our skin. Actually, something like 1/3 of all our proteins are collagen, I think it's pronounced proteins. So you remember all these cosmetics, they always try to target collagen to support so that they stay rigid, so that the skin doesn't get old. Because at some point when you get older, the reproduction goes down, and that's why we look older, like we get wrinkles and stuff. And that's just because this protein is not that elastic anymore, and also the amount is smaller. And that's why all this cosmetics, they say collagen, but that's the reason. Okay, another interesting protein that I decided to put on the slide is crystalline. We find this protein in our eyes. When our eyes, they're normally transparent, right? If you look at people's eyes, then, well, they're very transparent. But sometimes, especially older people, like their eyes, they look a bit milky. Maybe you have noticed that. Maybe you've heard about cataracts, and this is the reason for cataracts. These proteins, they get damaged, and in healthy young organisms they get eliminated by the special mechanism. But when an organism is older and maybe not that healthy anymore, then these proteins, they start accumulating in the eyes, and that's why like the eyes got so milk is not transparent anymore. At some point, it's possible to just remove them from the ice. Other proteins, they are motors that produce energy. They are constantly in rotation, in motion. And the final example I have here are receptors that transport signals. When we have headaches, then this is the protein that is responsible for sending the headache signal from one cell to another. Okay, uniprot, swissprot. What is the difference between swissprot and uniprot? Very basic question comes in every single exam. So, what's the difference? Swissprot, uniprot. I know it's too trivial, but just say and then we move on.
This project is manually curated, so actual people are looking at proteins and...
At proteins, people who are employed by the maintainers of the database, they perform experiments?
No, I mean at the literature.
Yeah. And what is the information stored in there? Where does it come from? So one is manual, the other is automated. So what is the information in the automated part of the database? Where does it come from?
Many of them are predicted.
From where?
From other sequences.
What sequences?
From the correlated ones.
No, it's actually much simpler. So you're a biologist, you sequence a new organism, and then you run a gene prediction tool. Everything that it predicts goes in there. Yeah. So most of it is like true, but some of them can be also mistakes, but can also be true. But you know, the sequencing costs, they decline rapidly, and people sequence so much, but just like so much. And that's why this database actually increases super, super fast. Do you remember how much it costs to sequence the first human genome? Do you remember? $1 billion? When was it? Do you remember the year? 15 years ago. And what does it cost now to sequence a human genome?
I didn't understand.
What does it cost now to sequence a human genome?
1,000.
Yes. 15 years. That's a big difference. So, yeah, there are companies, they in the States, they can sequence your genome for, like 10,000 because they also want to make money. But the result will be delivered on your mobile phone really fast and nicely visualized and stuff. So sequencing is not a challenge anymore at all. But the manual annotation of these proteins, that they really, so the verification that they really exist, that's still old school, still people sit there with their chemicals and their microscopes, and it still is very slow and still costs tons of money. And in 2013, the gap was like that, and it's huge. It's obvious. It's big, and that is the motivation for us and you guys to be here to learn how to develop these computational methods that can close this gap between this and this. So you throw into this predictor this, and then you get as an output this. And as a matter of fact, I read a few years ago an article saying that for the localization problem, the prediction methods that were there, my tool not included, other localization methods that are there, they perform as good as experimental methods. So whether you send a person to do experiment or whether you run it with a predictor, the accuracy would be about the same. So that's where we were a couple of years ago. So it's really fascinating, really stunning. Okay, gene ontology. If we ask you in the exam, what is gene ontology, what would you say? Just try to come up with something. What is gene ontology? Someone? Just give it a try. Yeah.
Like something, I'm not sure how to explain it, but like,
Okay.
like a standardized vocabulary to assess not only like the function or the cellularization, which specific protein, but like also the relation or like the hierarchy. Well, not really the hierarchy, but yeah, like standardized vocabulary too.
That's absolutely correct. The standardized vocabulary gets many points. I would continue that describes protein function. And then protein function can be described by 3 different ontologies, groups, and these are these 3. Cellular component, biological process, molecular function. These slides are from 2013, so these numbers are outdated by 3 years, but even back then you can see that this number is like already big. It's very impressive. You would think that the cell, it's an entity like with a limited number of compartments, and then we see this number. It's impressive, I think. Another question that I have for you guys, and maybe each one of you, even if you don't want to say, but I still want you that you think right now of examples. Can you come up with each of you with one example that describes, that is part of each of these hierarchies? I'll just try for you in the head, because be sure we will ask it. And if you cannot come up with it, then make a note that you know for the exam later. And if you don't want to tell, then tell. What can examples be for each of these 3 ontologies? Can someone try?
We had at least one example mentioned in the exercise.
Guys, try. Computer scientists, they managed today. You can do it as well. So one example for each one of them. Okay, so altogether component, what can it be? Cellular component? Very good. Process, what is a process? Like? Localizes, binds, divides, grows, catalyzes, everything that is a process. Any verbs you can think of, they fit in here. What is a molecular function?
Protein binding, DNA.
Binding, that's a process. No, hold on, keep on going. Say more. Binding, yes. What else?
Transference.
Yeah, totally. Enzyme. Yeah. Good job. And can you give me an example of 2-level hierarchy for localization? So a term and the child term. Can you try? You can just make up one. It doesn't have to match the tree 1-to-1. Okay. For localization, parent node and the child node. Can you give an example? Okay, so give me a compartment, any compartment. Membrane, good. Sometimes there are 2 membranes in bacteria. So a child node would be intermembrane space. Okay, so we can also start with intracellular. Everything in the cell, intracellular, then the child node would be the nucleus. You see? Or it can be nucleus, and then the child node would be a nuclear pore. Get it? Okay. And this is a real example now, a real tree for just one protein. And this is to show that that's the structure of the tree as it can be. Not always like one way, just one way down. If there can be several, goes in different directions. But in the end of the day, it's always a directed graph, and there are never circles in Go. But you know it because you work within your exercise on that. Alright, so we talked about eukaryotic cells and prokaryotic cells. The difference between them is that these cells are compartmentalized. They have organelles, while these cells, they don't have organelles. Okay, they have membranes. Sometimes they have two membranes, but inside the cell, this is just like one entity, and everything is just swimming there. The DNA, all the proteins, they just swim in this gel-like structure. Then there are these things, these hair-like structures. They help bacteria to move, but not all have them. And prokaryotes, they are archaea and bacteria, right? Archaea, they are even simpler structures. So these cells, they are about 2 billion years older than our cells, and they are accordingly simpler. Maybe you know that mitochondria, they are remnants of bacteria. You know that? At some point, eukaryotic cell has swallowed a bacterial cell, but for some reason decided not to digest it, but decided to keep it. And then bacterial cell proved to be useful as it provides energy and phosphate. And mitochondria is the only organelle with its own DNA. Have you heard about this case that happened maybe like 3 months ago when there was a child born from 3 parents? No? So the third DNA came from mitochondria. So the case was that both parents, they had, I think they were either deaf or some other disease. And in order to produce a healthy child, they just got a donor who provided mitochondrial DNA. And then the child was healthy. And so nowadays it's possible to have a baby from 3 parents. That's the way. That's the reality. That's where we are right now. It's future already. I think it's awesome. Okay, so we are interested in localization because we can assume that localization gives us information about physiological function of proteins. If a protein is localized in the nucleus, we can say that, okay, it is not involved in energy production, for example, because it's in the nucleus. Yeah? Subcellular localization is super important for drug discovery and drug production. This chart shows that what are the compartments that all drugs that are out there right now, well, 2008, target. And we can see that something like 80% of all drugs that target either extracellular, cytoplasmic, or membrane proteins. Can you explain why? Why do you think this is the case? Why everyone develops drugs to target just these 2 compartments? Why do you think this is the case? So if you think of ibuprofen or aspirin, the drugs that prevent us from having headache, right? They target membrane proteins. And many other drugs do. Why? Why these proteins?
I think that the membrane proteins are in charge of like trafficking across the system, across the compartments, and so I think it's important to regulate this.
Totally, that's where this huge junk is coming from. Totally, yes. What about these guys? Like, what is the rationale behind developing drugs with this instead of like sending everyone to the nucleus, for example? What do you think? Well, if you think of the topology of the cell, does it help you to answer this question?
Because it's easier to target an extracellular protein.
Exactly. Yeah, it's just the accessibility. Because a drug, this component has to go through the cell, inside the cell it has to find the right compartment, and then their performance function. Who knows if it's going to work this way? It's much easier to target proteins that are in the extracellular and in the cytoplasm. How much does it cost to develop a drug? What do you think? I think you will talk about it later at some point, but that's a rationale why it makes more sense to develop drug easier way than to develop a drug that would be maybe more effective, but it's riskier. So I wanted to give you an idea of why it is so risky, why people would rather prefer to develop this kind of drug than this kind of drug. So drug development costs how much for one drug?
$1 billion?
Yes.
Because
Yeah.
most are not.
This is like ballpark, billion. How long does it take? Like what is long?
10 years?
Average is like 11, 13, I think. And how many drugs are being approved each year, like new drugs?
20.
Um, Let's just talk about the states, one country. What do you think? Oh, what is your assumption? Like, how many new drugs are being produced every year? Not produced, allowed to be sold. Just give a number.
I think it was 20 worldwide.
20 worldwide, one country, then how much?
One.
Exactly. It's like 1, 3, 5, sometimes none. So this research goes on and on and on. People develop drugs, they research, but in the very end of the day, that there will be just very, very few, less than a handful of drugs that will be accepted. So all this research, all this money, everything, and the winner is going to be just like very few. And that's why it's also so risky to invest all this money into something, do this crazy research. It's just very, very expensive. And that's why people are interested in that. And this table here shows that localization is actually one of the key points that people consider when they develop their drug, in addition to other properties, of course. Okay, so this table shows us examples of drugs. Some drugs that target certain compartments. Again, we see plasma membrane, we see, okay, the rest are organelles, but this table shows that, okay, there are diverse drugs that go into different compartments, and they are there for things like, yeah, cancer, some other diseases. Yeah, so localization, knowing how to design something, how to send somewhere, it's like one of the main aspects they consider when they develop these medical drugs. And this table shows that NLS, this is nuclear localization signal. This is a signal that sends a protein to the nucleus. It's just like one fragment of a protein that is responsible for doing that. And a mutation just in that region can lead to a whole range of diseases, and then they're like really, some of them are genetic diseases, some of them are acquired diseases during life. But once the protein has some kind of mutation and cannot reach its correct localization, then diseases like all kind of types of cancer and so on can emerge. So we know from biology, from experiments, that once there is a mutation and protein is not localized correctly, we'll expect something like that to happen. So they're linked. Diseases and mislocalization are linked. Can you recognize what's happening here?
Cells are dividing.
The cells are dividing. What does tell you that they are dividing?
Because like the picture left.
This?
No, the left.
This.
Yeah, it's like you've got the in the, how is it called? Like metaphase.
Mm-hmm.
Something, and like
Cool.
it's the chromosomes that are being pulled apart.
Super. Excellent, yeah. So this is the normal division of a cell. They form this on both poles. The centromeres, they pull these fingers that pull this cell apart. And then the chromosomes, these are not chromosomes, but they're inside. They're split between the two. And it's a symmetric, beautiful structure. And then in the end we have these two beautifully separated cells. That's how it should be. However, if there is a mutation in a protein called like that, just one protein, cells, they stop dividing, they try to divide, but they do not separate. And this leads to something like that, where we see that these nice structures, they're not symmetric anymore, they're like all over the place. And what is the result of this? What is it? How do we call it? This disease when this happens. So cells, they're not dividing anymore, they are accumulating into this aggregates, into this bulky big structures, and they keep growing. What is it? You all know, I'm sure. It's a disease. Many people have it. A cell cannot divide, cell starts growing. Because it wants to divide, but it cannot, and that's why it's... What is it? It's cancer. So this is cancer, and normally cancer cells, they occur in our bodies constantly. Normally they're being immediately detected and they're recycled and stuff, but sometimes they escape this machinery. Yeah. So one protein, crazy things. Okay, so just to recap, why localization information is important? Well, because it tells us something about the functional role of a protein, because we said if it's in the nucleus, we can assume that it does not perform, I don't know, energy production anymore. Localization is super important for drug discovery. Aberrant localization, this means that if protein is mislocalizing, then it's very likely that a disease can occur. And finally, knowledge about localization can help us to analyze protein-protein interactions. Can you think why this is the case? Why does it help us to say that the proteins are interacting or not? Why do you think this is true? Yeah?
The different compartments, they won't be able to interact.
Yeah, exactly. They will just never meet. Like if we have one protein sitting in the nucleus and the other one sitting in the mitochondria, like they have no chance. Even if they want to, they cannot meet. And how many are potentially possible protein interactions are there in human? How many proteins are there in human? How many proteins are in human? How many? I heard 100,000?
I said 20,000.
20,000? Yeah. Yeah, 100,000, that was the state of the art 15 years ago when the first genome came out. They ran this program, a gene finding program said 100,000. Everyone was like, ooh, cool, we are more complex than the worm, and then the Arabidopsis, and then corn. We are complex beings. It turned out the number is way lower. It's like 20,000. As a matter of fact, it always like changes dependent from the release of Swiss prot. We assume it's something like 20 to 25,000, but the exact number, that's what the Swiss prot tells us every month. So that's state of the art, and that's 20,000, yes. Yeah, so the number of possible interactions, that's like 2 out of 20,000. Huge number. Yeah, you remember binomial, 20 over 2. The result is like factors. Big number, so if we consider localization notation, then we can limit this number like incredibly. Now, Why localization predictions indispensable? Well, because experimental methods, high throughput methods, they cost money, they're not accurate, means that you can always make mistakes when you run experiments. Either you can, as a human, you can mess up with, I don't know, your chemicals, you can mess up with your temperature in the room so that no one else can reproduce the result, or if you do everything, you can mess up while documenting your observation, or even if you do everything absolutely perfect, then someone who curates the database can make a mistake while putting down the annotation. So this is the human factor, and the annotations and databases, they're never going to be 100% correct. Impossible. It's also the case that people, they run experiment, they see something, they go, okay, this is annotation, then someone else does it and he sees a different result, and then it can be that the first person just didn't see the second result, so they also change all the time, the annotations change. They get removed, they get merged, that's what happens. So they are not 100% accurate. They are not complete. Why they're incomplete? Well, because people, we talk about, for example, when they develop drugs, then they're most interested in extracellular, cytoplasmic, and membrane proteins. They don't care about other proteins, so why to study other proteins? And other people, they go like that. They would study only their proteins of interest, and others, who cares? Why spend money on them, right? And therefore, the databases, they're like super biased towards certain organisms or towards certain functions or localizations. So there are problems, and this guy, this is the yeast, Baker's yeast, is a great example for why experiments are not perfect, because yeast, this is, as you all know, is a one-cellular yeast, right? It's eukaryote, it divides super fast, so you don't need to run an experiment and wait until a mouse gets pregnant and then carries the child and blah. So it, you can see the result like immediately already with the next generation. That happens within milliseconds. It has been studied for 2 decades. All proteins are annotated, all of them. However, localization we still know for only 2/3 of the organism. This is just not trivial. This is just not easy to do. That's why people need us. Okay, localization prediction is a super cool task because it's easy, the easiest one when you predict function, because the number of compartments is limited, so you don't go into unknowns, you know what you're working with. The annotation is clear, somewhat, I guess in comparison to other tasks it's clear. Most proteins, they're active only in one compartment, so that's cool. It makes your task easy. And finally, the process of how proteins travel, it's well understood. We understand how it works. We can describe it with a schema, and this also, of course, helps to predict it. So it's a very nice playground, also for master thesis. Okay, so this trafficking in the cell, as I said, it's an understood process. The key message here is that there are 3 ways of how it's done. Either all proteins, except of mitochondrial, of course, are being produced in the cytosol, then they can travel to the nucleus or back through the pores. They can be brought by other proteins to other compartments. That's called transmembrane transport, because they have to pass their membranes. Or there is a third one, the vesicular transport. That's when the protein gets into a vesicle, and vesicle, that's like an autobahn that brings the protein to its correct destination. So there are like autobahns for the cells, like veins, that move the trouble through them to these compartments. And we know, for example, that the protein needs to go to the cell outside. It has to go to ER, it has to go to Golgi. Sometimes it goes to here, and then the damage is just there. So it's like really a model. Now, sorting signals. I'm accelerating now because I see that we have not so much time anymore left. Okay, sorting signals. Okay, so sorting signals are those signals, those regions in the sequence that tell the cell where protein needs to go. This part of the protein can be responsible for binding, cutting, accelerating, catalyzing, I don't know what, while this part is going to be responsible for localization. So some other proteins will recognize it, will see, this localization signal, I bind to it, and to this part, and then with this interaction, it's going to bring this protein to its localization. That's how it works. Sometimes the signal they're distributed across the whole sequence, and only if the protein comes together in its three-dimensional structure, another protein can recognize it, and only then it binds and like takes it with it. So identifying this experimentally is tough, because in order to identify where the signal sits, you have to mutate every single amino acid and see whether protein changes its localization or not. If you change here amino acid, then of course you will see the correct localization, and you say, this is not my localization signal. But if you change here and then the protein does not go to the nucleus, if it's nuclear, then you know, it's part of my localization signal, then you keep on moving. So super tedious process, like lots of work, and doing that, it's even worse. Because even here, if you're limited, you know, you can assume it's going to be like 30 amino acids long. Here you have like no idea where it's going to be. So of this we know some, of this we know less. That's the problem. So that's where we stand with the sorting signals at the moment. As a matter of fact, there was a Nobel Prize given for the discovery of this signals in 1999. It's the researcher who sits in New York, Rockefeller University. Burkert knows him. So he said the signals, they're important, they govern the transport in the cell, and I'm going to call them zip codes. So this localization signals, they're zip codes of the cell. So the zip code tells where a protein is going to be. It's going to be in the eukaryotic cell, in the intracellular space, it's in the nucleus. Okay, we don't know where exactly in the nucleus, like what house number it is, but that the localization is the same. It's in this signal. So signals are zip codes. Key message for the exam. Okay, in 2013, there was another Nobel Prize given on this topic. It's about the vesicle transport, and this here is to show, just to show how valuable this localization problem is, even though people work on it for 20 years, and then there is me who develops the 30th method for localization prediction, it is still valuable. There is real value in developing this machine learning methods for biological problems. Even if you think for later your PhDs and master thesis that, well, people did that already, why should I improve on that? You should. The whole point is that the information changes. There is always more information. The information becomes more complex. 10 years ago, people didn't have the technology people have now. And now you can just get more insight and get more value. So go for it. And it's going to be recognized. With my method, I published it 4 years ago. It became state of the art, like immediately it was better than anything else, like significantly. And 4 years later, there is still nothing better. And you publish, you go to conferences, you present. It's awesome. All right. So as I said, people were studying localization for a long time already. And that's how they were using their method. So they were looking for the sorting signals. If they saw a sorting signal in the sequence, it was on one of the sides, they were like, okay, this is likely a localization signal, and always it worked. A great approach, a very simple one. The problem is that we don't know this localization signals. Well, we know some, but not all. So we're going to miss a whole bunch. Yeah? Okay, other methods, they said, I have a sequence, I don't know from human, I don't know where it's localized, but if it is homologous to chicken from nucleus, to rat that is in the nucleus, to, I don't know, a dog that is in the nucleus, and a monkey that is in the nucleus, so we can assume that our protein of interest is also in the nucleus. This works awesome. The problem here, DNA, but only very quick, because we're out of time. It's important to understand the concept. So what is the problem with this approach? If it works, it's great, it's the best. But there are problems. What are they with this approach?
You don't always have that much info, actually.
From info about the homologs? Yeah. Oh, you don't have the homologs, exactly. So this one is your problem. Eukaryotes, the number of eukaryotic genomes is lower. We can For most proteins we do have homologs, but they're not going to be annotated always. For bacteria it's way crazier. We have thousands of bacterial proteins, and a whole bunch of them just have no homology, like nothing. So, works sometimes. Next one, text-based analysis, is another super cool approach. People go to PubMed abstracts or PubMed papers, get information from there. Imagine you publish a method, it gets published today, you say protein is nuclear, you cannot assume that this information will be used for tomorrow, right? It takes time for the paper to be discovered, to be read, blah blah. So this approach is very, very useful. What is the problem with them? Why cannot we annotate everything, the whole human, with these approaches? So you always have access to the latest publications, to the most current version of PubMed. But why can't you still annotate whole human? Any ideas? Well, you can only find information if the information is available, right? And if someone did not make an experiment for a certain protein, the information just won't be there. So there is no chance for getting something when it's not there. Make sense? Okay. No? Yeah? Okay. So great methods with drawbacks. Then there are us. We do de novo. De novo means from the first principles. We say, Burkhardt says, forget about all that, all additional information. The nature, biology also don't care about PubMed and homology and signals. They don't use this information to make us functioning. All the information the biology or the nature gets is already in the protein itself. It's in the amino acid sequence. So why not to explore the sequence just to get this information the way as biology does it? So this is the de novo approach, and we play biology by applying intelligent machine learning algorithms. And then there are hybrid approaches that combine any of these methods. All right, so problems to consider. And this is the recipe of machine learning now. Don't matter what you're going to predict in the future, whether it's going to be a biological problem, whether it's going to be a human behavior problem, whether it's going to be a problem whether airplanes fly, I don't know what. The recipe of how it works starts here, and it's, believe me, always the same. The principles are always the same. So first you need to think of your data. Where does data come from? Can be private source, can be open source, right? Your data will always be noisy. Don't be naive, thinking, oh, I have best data from best experts, I can just use it. No, you cannot. It's going to be dirty. For biological data, the dirtiness comes from the biological, experimental, and curation levels, as I said before. Human factor, always there. You cannot really get rid of it. It's dirty. Then, when you develop a method, you have to make sure that you assess the performance correctly and properly. That you eliminate sequence similarity between training and test. If you do not do that, then you will predict just what you are trained on. You will do 100% great, but if you apply your method to a new data set, like, you will fail, right? Right, then public databases, as I said before, they're super biased towards certain proteins, towards certain problems, towards certain classes, so don't trust them either. Keep in mind that data is created by humans, not by super beings, and humans, they only do what they need, not more than that. That's the reality. When you develop a method, make sure that the method is useful in the way that people can understand what it actually prints out. Sometimes we ask students to show us results, and then the students send us an, I don't know, XML file of this size, and then we mentors have to find one line of the result. We go like, this is not useful, please send us another email. Just think of it in advance, just make the life easier for the mentors first when you write your thesis, and for the users later. Just make it easy for them to understand what you actually deliver. And finally, benchmark. But benchmark, that's we can talk about it later. All right, so that's the method from the master thesis called log32. The idea here is that you use the sequence and then you use the knowledge of how biology functions, and then you try to implement it for all possible sequences, for all possible domains of life, so that you can really close the gap, like for everyone. That you develop a method that is useful for everyone, no matter what people are interested in. They can just use that and they will be happy. Some people develop methods only for humans or only for yeast or something. That's great, but the application is limited, right? You want to be known, so develop something that is like Google does it. Everyone can use it every day, 10 times a day. So, a joke. It was a joke. Okay, so this method log32, so that was one framework for, as I said, all proteins. It was robust for sequence errors. I will tell later what it is. It predicts 3 classes for bacteria, 3 for archaea, 6 for bacteria, 18 for eukaryota. These are localization classes. The numbers, they seem low. If we think of the Go tree, there were like 3,000 something terms. These numbers are low, but this is the best. This is as good as the number of classes that log3 predicts is as good as double of what other methods predict. So that's as far as we could go in 2012. Only these classes. For the other classes, there were just not enough information available. Imagine that. Okay. So we use decision trees to resemble this graph. We provide scores that tell whether prediction is super reliable or not so super reliable, and the performance, of course, was also good. Okay, now, so this is machine learning as it is. What we do, we prepare our data set. We train a method, we test a method, we always compare ourselves to external tools. No one knows how precise we did this. No one knows whether we lied, how true we were when we were doing this. So no one is going to debug our programs. But what they're going to look at is how you perform here. So here they have just trust you, say my performance is this and that. Trust you, I have no other choice, but I want to see how you perform on something, on a data set that other methods also use, and on this will be like your true final performance. Right, what do we do if we don't have other methods? How do we still assess ourselves such that people can believe that our performance is true? So ideal case is that you train and test, you report a performance, like 60%, and then you expect that on some external data set, when you compare yourselves to other methods, the performance will be the same. That's what you expect. You don't want it to be super high, or you don't want it to be super low, right? You want it to be somewhat in the same range. But if you don't have any other, say you predict for the first time ever whether protein is responsible for developing Alzheimer's disease. There are no, let's say there are no other tools. How do you compare yourself? How do you do this step if there are no competitors?
You compare with yourself, and like you use different training sets. And then
different training sets? Why?
Like split your data and tools.
So you develop on 9 sets and then you develop one more time on one set, or what?
Well, for example, you can have like an independent set and then 2 sets, and you train on one set and you perform like your method on the independent set, and then the second time you do the same, and the results should be like quite the same.
So you train twice?
No, once.
How once? You test twice?
Yeah.
Okay, that's one approach. Cool. What else? Okay, you always compare yourself, no matter what you do, you always compare yourself to random. You always want to be better than random. That's like your baseline. Okay, so data set. As I said, all we had 4 years ago were proteins for these few classes. That was the reality. This was the number of proteins. Right. So we'll talk about that. Okay, so that's the resource that you also all use, that's UniProt. You extract all the proteins that are experimental, annotated from a text file that looks like that. So you have protein names, you have localization annotation, and you have a sequence. So easy. Then you have to do homology reduction. Homology reduction you do because the databases are biased, right? If you have in your database this much of nuclear proteins and this much of all other proteins, then be sure you will always predict nuclear proteins, right? You will never catch the small proteins. And therefore you have to somehow understand what the bias is and remove it always. There are tricks when you don't work with biological data, but maybe with other data, there are tricks of how to synthetically balance your data set, because ideally you want all your classes to be represented somewhat similarly. That's the ideal case. So anyways, in biology, what we always do, some of you guys have already worked with us, so you know, I think you all know from the exercises, you do homology reduction, HSST value, you guys know? Do we need to go into that? No, great. So yeah, and then you apply that threshold, and sometimes you also run plus, just to eliminate all, really like all homologs. That's how the statistics look like. That's what we got from SwissProt. And after applying homology reduction, these were the numbers of proteins that were left. Okay, now comes cross-validation. That's what we do with our data set. So here the idea, do you all know what it is? Can we skip it? Cool. Okay, so the idea is just to train on the whole set and to test on the whole set to maximize all your capabilities. Okay, so always when you perform machine learning, of course, you always want to train on as large data set as you can have. And in my case, I ran this range reduction, and you remember only very few proteins were left. And so how to maximize those proteins. Okay, you apply a trick, so that's what we have, training and test. They are super separate, super different, and there are just few, and I want to maximize my training set. What do? I go back to my old SwissProt proteins, and then for every protein in the training set, I get a homolog. So that's how I artificially increase the training set. So they are allowed to have homologs, they are not allowed to have homologs. Homologs of these guys, they're not to be homologs to test set, you see? And that's like the trick, maybe can be useful for you for the future. Make sense? Yeah, cool. Okay, performance estimates. As I said, super important. Accuracy. Okay, accuracy tells us how often predicted class is correct. So what we look at the number of true predictions divided by all our observed proteins for a particular class. Coverage tells us how often the observed localization class is predicted. So before we were dividing all true predictions by all proteins in the observed set. Now we do the same. We divide by all proteins in the predicted set. These two values are very important for the exam. Make sure you can explain what they are. Make sure you know the formulas. Okay. Now, you have your precision recall, accuracy coverage. Now you want to know how reliable is the estimate. The rationale behind this is that you develop a method. You say that the performance is 80%. Then 10 years pass, and then your performance is 85%. So the question becomes, did you lie? Did you underestimate? Or did you just forgot to report your boundaries of where your prediction performance actually lies? So sometimes when you buy sugar, I don't know, I hope this is true. I saw it in different countries. I don't know if this is the case in Germany as well. But sometimes they say like 1 kilogram or 1,000 gram plus minus 5 gram. Or I think chocolate bars also have it sometimes. I know that I saw it. So the weight can be a little bit different dependent on what machine is being used and so on, right? But it gives you the range. So everything between the range is acceptable. You go for it. Everything that's outside is not acceptable, like you're being cheated. Here it's the same. You report performance and you give an estimate within what it's still acceptable. So if we apply an independent test set within this range, we accept it to be. So if you send me as a reviewer your method and they see, you performed within a standard error, great, pass. If you don't report your standard error for me, I will tell you, no, not going to be published until you show me what your standard error is, until you show me what's your deviation. Otherwise, I'm not accepting your method. That's the reality. So, important concept. Okay, 15 minutes. What is the way of establishing the standard error? So standard error tells me I have a performance, say it's 60%, right? And then I say that standard error is plus minus 4, let's say. So 61 acceptable, 62 acceptable, 63, 64, 69, 58, 57, 56, right? It's within standard error. Where do I get these values from? Okay, I'll give you a hint. For that, you need another data set. Right? So to estimate the variation. But where to get it from? It's very tricky question, I know. But it's part of the process.
Randomly take a fragment or a fraction of what you have, then you predict on that, and you average the accuracy.
Super cool. Yeah. Instead of re-predicting, you can just do bootstrapping. And bootstrapping was first introduced in Baron Münchhausen. If you remember the book, the guy, when he fell into a swamp with his horse, he just like pulled himself by the hair out of the swamp, and that's the way how he rescued himself. That's at least what he says. So that was bootstrapping, helping yourself with all means that you have. And so in our case, what we do, so if we have, say, our predictions, say, I don't know, plus minus minus plus plus minus 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 plus plus minus. So these are predicted localization classes, like nuclear, non-nuclear, and our performance on them is 64%. So in order to establish my standard error, what I can do, I can just get a subset from here on the predictions and estimate the performance on them. It's going to be 65. Okay, what I do next? I get another subset and I estimate performance on it. It's going to be 66. And then I take a third subset from the already predictions. So I don't need to even run any predictions anymore. They're already here. I just get the values, accuracy based on the subsets. And it's going to be 67 or something. And that's how it's done. This is bootstrapping. So they don't have to be consecutive. They can be like 2 can be from here and 3 can be from here. But the idea is that you always get a subset from your predictions, and then you evaluate your predictions based on the subset. So that's bootstrapping, simple. Okay, everyone got it? Yes. Okay. So that's more formal algorithm. You start with your set. You randomly draw a subset, you estimate your accuracy or your metric on the subset, and then you repeat it. And then you get your values, and then just apply the standard deviation and standard error formula. As simple as that. Okay, so we have the data set, we have the metrics, now we are ready to train the method. These steps were involved defining what the machine learning algorithm is going to be. Then I said it's going to be an SEM. SEM works with kernels, all right. So what should be the kernel, what should be the classifier, and how to optimize it. Always the same steps of machine learning, always. Okay, so what is an SVM? SVM, that's a machine learning algorithm. Do you guys know what an SVM is? Oh, cool. Okay, so we have feature map. That separates. Awesome. And the kernel function does the separation, right? From input space to the feature space. Wow. How do you know?
Look at the second lecture.
Wow. Very impressive. Okay. So I said, so first I was looking for different machine learning techniques. Some of you work with Becca for the exercise, and Becca employs over 100, maybe hundreds of different machine learning algorithms. I tried like a handful with basic settings, default parameters, default everything, and I found that on default everything SVM performed best. I said, all right, then we go for SVM. Thing is that the best way to find your best performing algorithm is to try all algorithms with all parameters, with all settings, and then it's going to take you 20 years or so, even in a parallel computing setting. You cannot do that. So you have to come up with assumptions with simplifications. So I said, all right, I started with basics for each algorithm, whatever works best at default, I go for it. Assumption that worked. So SVM worked. SVM can be used with different kernel functions. So I researched the literature for different kernel functions, and then I found the profile kernel to perform best. Profile kernel is an immensely powerful technique. After finding this out, we started applying profile kernel like as good as everywhere. You're also applying, I think, in your exercises, so that's where it's coming from. So what does profile kernel do? Profile kernel takes a protein sequence, and then it adds information to this protein sequence from the evolution of the protein. It goes into the homology of, you all went past, you all found homologs, and then the profile kernel was looking into how well is a certain amino acid conserved at each position in the homologs, and then this information, this like junk of homology of evolution information, was inputted to SVM. That's what we all do. Maybe it's hidden in your codes, but that's what's happening. So how was it done? We have two protein sequences. We have our profile file from our homologs that tells us, okay, these are all protein amino acids. This is our protein sequence. And this profile file tells us, I think you have created them even, tells us how well is an amino acid is conserved. Do you know them, these files? Profile files, excellent. Okay, so then you, as a user, define certain parameters. You also define them. And when you were running this kernel k-mer length and the conservation threshold, and what happens then is that this kernel function defines a vector of the size of all possible mers of the length as you defined here. In my case, it's 3, so it creates a feature space of 8,000 different k-mers. And this is what happens next. So for each 3-mer, so these are all possible 3-mers, and then for each 3-mer, it checks how well it is conserved in the profile. So it starts with the first 3 letters, 3 because of 3, and then it looks, this k-mer is conserved in this 3 letters in the following way. The first amino acid A is conserved, the first C, this is our sequence, these are k-mers. Okay, so the sequence, the first 3 amino acids from the sequence, the first C is conserved to 2, the second A is conserved to 1, and the third letter T is conserved to 1. So if the sum of these conservations is less than the threshold, then we increase the value in the feature vector that is corresponding to this k-mer by 1. And then we do this comparison for all pairs of 3 amino acids throughout the whole sequence. So this vector can have also other numbers. And this we do for all 8,000 k-mers. We do the same for the second sequence, and then the output would be the multiplication of these two vectors, and this would be a simple number. So the larger the number is, the more it tells us how similar the protein sequences are in terms of their evolutionary information. So it's not just counting amino acids that they're similar between the protein sequences. So this is not homology in that way. This is homology that goes like beyond. It goes back into the evolution. That's super powerful. Okay. So what I did next, I compared the different kernels. I saw purple kernel performs best. So of course you go for the winner. That was the winner. Then there was a question, okay, do we have just, so for eukaryotes I had 18 localization classes. Now the question was of how to design my method to predict all these 18 localization classes. One way is to have 18 predictors. One predicts nucleus and everything else. The second predicts mitochondria and everything else, and so on. That's one way. Another ways are like placing somehow differently, randomly. So this approaches, or you're being very smart, you're a student who like pays attention to all details, and you think, but there was a Nobel Prize given like to this identifying this structure. Why not use the structure because you're a good student? And then you implement your trees that follows the same structure. Remember do you know what I'm talking about? That structure, the system with 3 transportation ways, gated transport, then to transport to other compartments and then to the outside. Remember this? So you use this information and then you try to build SVMs based on that information so that you send your sequence just through the same system but with SVMs, with machine learning. That was this number 5. So this is a multi-class classifier, 1 against all, 2 against all, and so on. Very useful approach. Then there are other ways of how to structure a tree, or you just do what I said. You implement this architecture in a tree of SVMs. So you're trying to basically mirror this into this. The only difference is that here I decided to discriminate between membrane and non-membrane already at this stage, and then if the first SVM says this is membrane, then it goes to the right. If it's not membrane, it goes to the left, but the rest is like really inspired by this. That was cool. Seeing that something that is done randomly performs worse than like what you implemented when you mimic biology. Cool. Okay, so that's how it works. 5 minutes. 3 localization classes. You send your sequence to the first SVM. It says either cytoplasmic or something else. If it's something else, then the second SVM says this is membrane or extracellular. As simple as that. Okay, so visualization for bacteria, same way. 6 classes, for eukaryota, 18 classes. Okay. So first, as I said, it's separation between membrane and non-membrane. Then next level is secreted, non-secreted, and so on. And this table just shows, again, the slides are old, I'm sorry, they're for 2013. My deck now would be more informative because this is actually not so relevant anymore. But it's an overview to show how much better my method, we call it MyND, was better. I see that it was as good as the others. Huh. Okay, then there was no difference, but it was a big difference to 1 against all, for sure. But this one was faster, because these were randomly built trees, this was just 1 tree. All right. That's not so relevant. Okay, that just shows that I was super faster than the others, 30 times faster than the others. Okay, so we trained, now we test. Cross-validated, it was 65%. 65% sounds low. not 100. If you develop a method with 100%, be sure that everyone is not going to take you seriously. 100% that's not possible. It's impossible. One reason is that just because the data is dirty. So develop something perfect and dirty data, that's already something. There is a problem with your code. And another one that 100% it's impossible. You're never going to reach 100%. So 65 sounds okay, but if we think about random, if the classes were distributed equally, then random would be 100 divided by 18, something like 5% or something. So way above random. All right. At first level, the accuracy was 94%, and it turned out that the best performing method that is out there that predicts membrane, that differentiates membrane from non-membrane, performs just the same accuracy. That was already impressive. Burkhardt was happy. And then the performances for different classes, they were good, especially for classes for which there was a lot of data. For classes where there was not so much data, the performance was accordingly lower, but still always above random. Okay, so this profile-based kernel, that was our finding of the century of this lab. We continue using it a lot. It's super useful, so maybe just the idea for you to remember, like it's cool going back into the evolution. Okay, now we compare it to other methods. There are other methods that use all kinds of different information, like amino acid composition, secondary structure elements, other SVMs, Go information, motifs, or they use k-nearest neighbor based on some other features. So there are really, really many other methods out there. And then there is us. And then we see that in comparison to other best performing methods, we're like, so this is Loctree 2, we look at Loctree 2, we're like better. So this is the number of classes. The reason why these numbers are 9 and 5, that's because these classes, they predict only 5 and 9 localizations. So we had to trim ourselves back to this 9 or 5 classes, and everything else that we predict we just discard, because otherwise we cannot compare ourselves to them. You get it? Yeah, and still, even on this few 5 or 9 classes, still we are better than them. We also compared ourselves to the others by tweaking our sequences. Namely, we were removing 30 amino acids from each of the ends and randomly removing some of the amino acids. The rationale behind this was that, well, sometimes you do sequencing from metagenomic samples, and then you do assembly of your configs, and then sometimes you make mistakes. Some part can be missing of your protein, or I don't know, what other mistakes can happen. So sometimes information is incomplete. And if evaluating the methods on incomplete information, then our method was like much better, significantly better than the others. So this evolutionary thing was so strong, so solid, that even if a protein has some kind of mistake, even it can be mutated, the mutation happens, but the Loctree method still captures the localization, while the other methods, they may already not find it anymore because of the protein mutation. Okay, so this thing of using the kernel, of using the tree, for incorporating additional biological information was very, very powerful. The tool was great. We actually went on and developed a next level tree for another kappa competition, for the second competition that we participated in. Our students, they first ran BLAST in order to find homologs. They said, okay, if a homolog is available, we take its annotation. If a homology is not available, we take Loctree 2. That became a method called Loctree 3, where performance increased to 84% or something from 64, so a huge increase. And in the second kappa challenge, this localization method was among the best performing methods. I think it was number 2 or number 1 even, or something. But just by plugging homology is super powerful. Homology is there. By plugging homology, we could improve the method like significantly. And another interesting part, of course, many thanks to Burkhardt, my master thesis supervisor from then, Bobby Hump, and the whole Ross lab. They were awesome. And then maybe one more thing to tell is that this is to show the bias of annotations in databases. So for all organisms on this slide, you can see that there is a huge bias for nuclear proteins somehow, for cytoplasmic, plasma membrane, and extracellular proteins, because that's just what people are looking for in their organisms for their needs. And if we compare annotations between, so inner circles, these are Swiss protein annotations that go crazy. They're like always look different, even for related organisms. But if you look at log3 annotations, then we see that these proteins actually are not so different anymore. Like human and orangutan, they're pretty the same for log3, but for Swiss prot, their distribution of their proteins is like much different. So that's the message to take, that databases, they are not capturing the biology as it is. Okay, and this slide might be a bit messy, but it's to show this. Here I compare Swiss prot to other localization prediction methods, and this just to show that no matter what method you're going to apply, you will never get just one truth. You will always get many truths, and then as a user, that's like your job to understand the middle, where the middle average truth actually is. Because look, for example, this is just an example of fish. I don't even know what this localization is, but you see that different methods. Well, Swiss prot predicts something like 20%, while other methods predict way less, and they predict very different numbers for this compartment. And then, well, something where they overlap is going to be most probably very much true, but then like, what is the other truth? And that's what we are, that's what we have, and that's how all our methods actually function in bioinformatics. So that's it.